of our investigation and our experiment, we established the next aims. The first one is to explain how the proteolytic enzyme works through an experiment. The second one is to identify how the peptide bonds reacts against the bromelain enzyme present in a piece of pineapple. The third one is to compare between two pieces of pineapples with a different temperature, which one will affect the gelatin surface. And the fourth one is to identify how the temperature of bromelain affects the pineapple enzymes. In these ordinary ideas, the research question of this investigation and experiment is in which way the enzymes in both pieces of pineapple affects the peptide bond structures, taking into account the temperature of each pineapple.
The equipment and ingredients that we are going to use are fresh pineapple, boiling water, gelatin package, glass or beaker, a spoon, pot, stove, glasses, gloves, and lab coat. First, we need to put 2 packages of gelatin to the container and add 700 milliliters of boiling water. Stir the solution until the gelatin has been dissolved. When the gelatin has been mixed, place it in the fridge and leave it there until it becomes a semi-solid. While the gelatin is in the fridge, fill a pot with water and put it on the stove on high heat. Once it's reached, add a couple of pieces of pineapple and let boil for 5 to 10 minutes. When gelatin has cooled as a semi-solid, we are ready to run our test. In the first test, we are going to place fresh pineapple on the gelatin and we are comparing it to our control. In the second test, we are going to compare the fresh pineapple against the cooked pineapple.
The results from our laboratory were the following ones. As you can see in the photo, the cup with the fresh pineapple seems to liquefies the gelatin, making its surface look clear, and that means that it breaks the peptide bonds containing on the surface of the gelatin. In the case of our cup with the boiled pineapple, we can see that the surface turns slightly clearer, making it seem somewhat transparent, but it doesn't break down the surface and it doesn't liquefy the gelatin, as we can see when we compare it to the control cup where not pineapple was added. This clearly shows how the bromelain enzymes present in the pineapple in the fresh cup affects the surface of the cup, of the gelatin.
Hi, my name is Mayra Barbosa and I'm going to talk about the conclusion of this experiment. So first of all, we must know what means the bromelain. So this is a mixture of different enzymes that are located in the pineapple and in which digest the proteins. So this is called the proteolytic enzymes. Proteo means proteins and lytic means lysis. Lysis is to break down something. So these enzymes are in charge to break down the proteins. Okay, knowing this, we can start analyze how each piece of pineapple acts at a different temperature. So we have the fresh one and the boiling one. The fresh one, its bromelain can work without problem because its temperature allows it. So the bromelain is an enzyme that has a catalyze and accelerate the reaction. So this unwinds the proteins of the jelly that is good with collagen, that is an animal protein. So this unwinds it, and that's why the piece of pineapple can pass the surface of the jelly. And the boiling one in the other side, the bromelain is disappeared. This is caused by the temperature because it's so high and the bromelain can't support this, so it disappears. And that's why this piece of pineapple can't pass the jelly surface. So this is the conclusion of this experiment, that the pineapple contains the bromelain, and that the bromelain depends on the temperature, because at high temperature it can disappear and its function it can be lost. So thanks very much to watch us and watch this experiment. I hope you enjoyed, and thank you. Bye bye.